to share with you my work, which is all about working together to understand the complexity of biology, with the understanding that, so, I mean, you mentioned my work with Günter Blobel. I mean, one could say, why are you, and the fact that now I'm primarily working on bioimaging informatics, and, you know, I've actually not, unfortunately, not been working on anything on the bench for a while. But one could say, why are you doing this? And really what informs my whole work has been the concept that in order to really understand the fundamental aspects, the fundamentals of biology, like I learned with Günter and at the Rockefeller in general, we really need to build technology. And I think like the same way that Günter and the pioneers of cell biology developed you know, a subcellular fractionation and EM, and none of what we know now would have been possible without developing the technology. My idea is that right now, one of the real issues that we have to address is the ability to share data across multiple sites and multiple collection points, because the complexity of biology can only be understood across multiple labs, and we cannot really have data silos, which we have right now. So in order to what I'm doing could seem separated from fundamental biology, but it's really informed by a thirst to understand fundamental biology. And so and I've been extremely fortunate since 2019, as you said, to be funded by Chan Zuckerberg as an imaging scientist. This really has allowed me to really expand all of the community building work that I had been able to start, but that really has really expanded enormously thanks to their funding. So now the work that I'm going to tell you about really is community-based and starts from, so I'm not going to tell you much about the work that I've done on integration of particle tracking and image analysis. So I'm going to tell you not much about that at all. But what I'm going to tell you is work that has started from the 4D Nucleome, which is an NIH-funded initiative, and then expanded to bioimaging North America, and now Quareblimi to really expand on the work that had been conducted since 2003 by the open microscopy environment community. So all of these different logos here are really integral part of the work. So they're not just here for display. And I'm based at the UMass Medical School, as you were saying. So the idea is that we really want to have microscopy reproducibility, but it has to be done by people that actually are able to do their experiments every day. So this has to be for the real world. I mean, everybody wants to do reproducible science, of course, but how can we do it when you're in the meantime doing all sorts of very complex experiments? So, the idea is that we want to build tools, specific community specification, but tools that go with those community specifications. So okay. So my work is then, my talk is going to be then about going around three different aspects. One is, why do we want to do reproducible science, and what do we need to do, and how can we actually do it for the real world? So why is quality control documentation and reporting important for imaging experiments? So there are three lines of answers to this question. One is, we want to organize our data. So all of us as on the bench has found ourselves with the frustrating situation of having to publish a paper. Often the paper is published maybe sometimes even months or even maybe a few years after you did the experiment. So where is that data? What are the conditions in which I acquired these images? This is something that all of us have found ourselves in. So we want to organize our data. We want to ensure quality, rigor, and reproducibility. Obviously, we're doing science, so all of this is central to what we're all doing. So this is obvious. And although it's obvious, but not so trivial to achieve when you're dealing with a lot of image data. And then we also want to do sharing value. And here by sharing, obviously, when we publish, we want to make sure that our data is available so that other people can take a look at it. But also, it is really important for us to access computational analysis, computational modeling tools, is that our data can be made available. So all those amazing people that can do this great data analysis can actually access our data and add value to it and go beyond what we were maybe able to do ourselves. So again, this idea that in order to really learn biology, understand biology, we need a team that goes beyond individual labs. So the sharing value is more like a collaborative value. So data organization. So this is actually an image that is available on Zenodo about the lost data map. I mean, I think this definitely resonates with me. I particularly like the Excel desert, the mountains of external hard drives. We have found ourselves in this situation. We have no information about the data, swamp of metadata. And instead, we would like to have something like this, in which we can easily find everything and know exactly what was done. So that is, I think, something that can resonate with a lot of people. And then there is increasingly, there has been already, like for a while, this is a paper from 2011, like reports of the difficulty of reproducing results. And for example, this is about actually from pharmaceutical companies not able to reproduce, for example, potential of, follow-up on potential drug targets because results were not reproducible. And so there has been studies done, for example, in this case by the Center of Open Science to actually evaluate this and indeed this problem with reproducibility has been documented. For example, in the case of, there are confounding information that has to do, for example, with the performance of the microscope that actually have been found to be related with the lack of reproducibility. And so all of us have this idea from like more or less anecdotal, but now there is actually studies that document this. This goes along with the fact that, there are studies now that shows how imaging methods are particularly underreported in even very advanced publications. So this is a paper that came out, I think, in 2019 or 20, in which people actually looked at publications in which that used imaging as their central or one of the key experimental techniques, and still had very little methods describing the experiments. And the one of the recommendation is we need accurate metadata. So we need, and I'll tell you what metadata means, but you have some sense, you have at least heard it, this word being bounced around a lot. But we need to have information about our data. And then another fear about like sharing data. I mean, is sharing data gonna take away from my ability to advance my career or not? Actually, no. I mean, there is, again, studies showing that the more you make your data available, the more impact it will have. Because other people will be attracted to it. There will be more results coming out of it, more citation, more impact to your work. So all of this is actually a win-win situation for science and also for increasingly for the career of people involved. Although, we have to still do a lot of work to make sure that people that do a good job in sharing their data are recognized. But there is stuff moving, for example, in the in the preprint level. This is a good example of things are actually moving in the right direction, and other signs of improvement. So what do we need to do this? I mean, we all agree that it is important, but what do we need? So we need, so this is actually an image that comes from a group in Germany, an FDI for bioimage, which hopefully will get funded. and start, putting more capacity building around all of this. But the idea is that we need research data management, in particular, fair research data management. Fair is the word fair comes from the fair data principle, which is findable, accessible, interoperable, and reproducible. So we want to make sure that our data is that way. And in order to do that, we need to work together in a bunch of different communities that are to develop various aspects, from standards and quality control to figuring out where we're gonna not only store and be able to retrieve the results that we have. So storage is one aspect, but storage is just the physical putting your data somewhere, but you can, if you don't have a good management of your data, then the storage is basically dead end. Instead, it has to be like you put it there so that you can retrieve it and you can connect it, connect the dots. That's the idea, that it's going to, you're going to hear about connecting your data with, for example, other type of data, the analysis that you have done on your data. And all of this has to be done with continual education and training for the users and a community building. So all of this is research data management. So, but a little bit more about what kind of value a research data management can add. The idea is that we have data that gets annotated so that you can do computation and analysis on it. If you just have data that cannot be understood by modelers, by analysis experts, then they will not be able to access it, and they will not be able to, for example, add value to your data. So the idea that annotation of your experimental data is really important for then leveraging at the maximum level that value of that data. So connects it out, and you want to make your research digital. So you want to annotate the source. We crack provenance means connecting the dots between where the data comes from and where it ends. So throughout the life cycle, know every step and take notes about everything, but do it in a way that it can be machine readable, so that then the data can be found and we can actually build knowledge. So the information that connects the dots is what we call metadata. So metadata, we've heard this word a lot, but it's essentially information that allows you to connect the dot between one piece and the next, and know what at each step what has happened. So it's a little bit like a treasure map to finding your data. So what does that mean? So we have, everybody is using either, a mixture of electronic lab notebooks and paper lab notebooks. These are typically mixture of metadata. So, when the experiment was done, what were the conditions, and results even could be on the lab notebook. But then if we produce data, images, these are data, but where is the metadata about this data? And so we need the metadata about the images as well. And so so we call that image metadata. This is an image that comes from the cover of Nature Methods, was done by Tao Do at the Allen Institute. And I think she was really able to capture this idea that all of these maybe geeky things, kind of not so cool things down under here, like maybe a little obscure. This is much nicer, right? This is not as nice, and these are a lot of some code maybe, but this is essential for us to actually see the picture. And so what are these pieces? There are pieces about the sample, how the sample was produced, how the acquisition was done, and the quality control, and the analysis. All of these aspects are pieces of the puzzle of what we need to know to understand really the results of an imaging experiment. But how can we do that? This is really difficult. Everybody's trying to do it, but it's not easy. So in order to be useful, this has to be really informed by three things that are constantly in our thoughts. It has to be usable. It has to be add value for the scientists, and it has to be as automated as possible. Not everything can be automated right away, but that's where we want to go. We want to make sure that things, whatever can be done automatically is done. So what are the kind of experiment information we are thinking about? So an imaging experiment is as sample preparation, image acquisition, and data analysis. And a lot of the work that we have done in the past couple of years with 4DN and Bina and OMI and Quareb has been to document really the microscopy metadata aspect. So that means the image acquisition. But very important is also sample preparation and image data analysis. All of these have to be documented. So in fact, in the I'll tell you more about the work we are doing with the microscopy metadata. But actually, before I going in the actual life cycle, before we get there, going along the data life cycle, I want to talk to you about a little some work we're doing with actually Claire Brown and Judith Lacoste at Canada Bioimaging and at UMass Medical School to actually document more than just the microscopy, but also the sample preparation and the experiment. So we're using Omero for that. And so what we're doing is the whole idea is to automate the import of images in Omero and connect and automatically incorporate the metadata, so the information that describe how the experiment was performed and how the sample was performed. And the idea is that we want to do this in a very close circle between development of tools and then for people to test it, and so that we can actually improve it as we go. And these two sites are basically our testing sites. From this point of view, it's basically just a single department, while this is actually the idea is to do it at the Canada Bioimaging level, starting from Montreal, but then expanding out. And so we are basically collecting metadata initially in Excel spreadsheets, in which we are designing together with the users the kind of information that they want to be automatically imported. And then we are basically the users deposit this information together with their images, and then they are automatically imported into Omero. So as a result, we have annotation in Omero of the images with information about the sample and also, in addition, the microscopy metadata. So now let's talk about the microscopy metadata. Microscopy metadata is about the information that actually documents how the image was acquired. And what we have done is a work that has really involved, expanded out from 4D Nucleome, Bioimaging North America, leveraging on the open microscopy environment work, and now really embraced by Quareb Limi. We have developed a set of specification called 4DN Bina OMI Quareb, so NBOQ microscopy metadata specifications that have been detailed, the kind of information that needs to be captured about the hardware and the settings for an imaging experiment. And these are but these are alone are not, they don't go anywhere. You need to implement them, and we are developing tools. Metal J2 is a tool that has been developed, that is being now developed by Claire Brown, but it started with Mark Sanders in the Thomas Pengo and Mark Sanders group. So they developed Metal J, and now there is Metal J2. And this leverages descriptions of the hardware built by another tool that is being developed by my lab called Micrometa App. So together, these two tools work really closely. And there is also a third tool that is called Omero MDE that is developed out of German bioimaging that is also very closely connected. Micrometa App allows you to document your imaging experiments in two ways. And this is, and you always have to think that Micrometa App is really bringing to life the NBOQ model, the 4DN Bina OMI Quareb model. So whenever you see something in Micrometa App, it means that it was developed by the community, and Micrometa App just makes it available for users to use. So there's a really close loop between the tool and the model. And what it first does, it allows you to document the hardware. And then this can be done by facilities or individual labs. And It is a little bit, at the moment, a little bit laborious because we're also working with manufacturers to make sure this is automated, but for now, a lot of it is not automated. Therefore, you have somebody has to go and hunt down the information. But once it is there, it can be used over and over again to document all the images that were acquired using that instrument in a second step. So basically, in a second step, what we do is we import metadata with bioformats from the image, import it from the description of the hardware, and then add further information in Micrometa app and generate a complete description of the hardware and the settings using Micrometa app. So here I wanna, what do we do with all of this information once we have? So first of all, I wanna emphasize the fact that this little thing here is basically to indicate that there is a model in which we describe an instrument with different components and describe different characteristics about each of the components. This model is being developed by Fordian Bina to extend the OMI model, and now is part of Quareb. But importantly, we are working with manufacturers to make sure that we are aligned with the manufacturers and the users are aligned in what kind of information can be provided and should be provided to ensure reproducibility. This information gets channeled into Micrometa app to produce essentially a description of the hardware and the settings. And then this can be used, for example, to document your images in Homero or wherever else you want to manage your data. It can be used to automatically create method sections using method J2. It can be, we are working, this is really more like work in progress. We're working to make sure that we can develop using this information. We're trying to look for funding to develop dashboards so that we can actually compare instrumentation across regions or even countries, or you know, for example, it would be great to do it in North America, South America, so that we can actually have comprehensive descriptions of instrumentation and share that information. And then importantly, there are now increasingly repositories that are being created, spun up. And this is a Bioimage Archive, is an initiative out of the VMBL EBI, which is actually open to the whole world to deposit image data. And they have published a set of very low level recommendation of what kind of information needs to be accompanying the image data sets. And importantly, doing it with Micrometa app more than fulfills the requirements, for example, for describing the acquisition aspects of the image data set if you want to deposit it in Bioimage Archive or and hopefully other repositories that will be developed. So if you do this kind of work upfront, and we're giving you, we're trying to work with manufacturer to make it as easy as possible, but then it will allow you to basically do a lot of different things that that you will, so that you are ahead of the curve. That's the idea. But one thing that I really want to discuss is about this idea of specifications. And, there has been attempts to do specifications in various fields. One thing that has to be really that has to be considered is, there are standards. Now we create a new standard, and now there are 15 standards instead of 14. The idea is that we really don't want to do that. We want to really build in a community so that every voice is heard at the beginning. We work together, and then there is a community-driven specification that considers all of the points of view. That's the idea. It's very hard, but we are, Quareb Lime has been extremely successful in attempting to do that. Quareb Lime has now something like close to 500 members from all over the world. And there are members from all sorts of different communities, and including, South America, North America, the rest of the world. Importantly, manufacturers and standard organization working together to actually build a consensus on what needs to be done, so that then whatever tools are developed take into account all of this. And one case in point that we are actually already doing, and it's been extremely successful so far, is working with key. So we are basically taking the overall model, which describes the instrument, the whole microscope, cameras, light sources, all of that. We're taking it apart, and we are going through each part with the relevant manufacturers. We are proposing the published model as our starting point, and then we are literally discussing every single line with them to decide what should be done. And so we have done this process already with we have done for around 6 months of talks with all of these representatives of all these manufacturers to come up with a revised camera model. And the idea is that what will this allow us to do, of course, it's not going to be immediate, is that it will allow manufacturers to provide the information about their instrumentation automatically. Ideally, both in the header of the file, of the image file, but also upfront. For example, if you purchase the camera, you could have the Quare Blini, the 4DN Bina Omi Quare Blini description of the camera in a format that then can be, for example, incorporated into a Micromate app, but also other tools that you may want to use. So the point is that Micromate app and MetaJ2 are two of the tools. Other tools could be developed. The important thing is that there is a joint description and that this is agreed upon with manufacturers so that we can actually achieve this goal together. So this is really, sorry, this talk has been shorter than 40 minutes, but maybe I've gone too fast. But but this is really a huge, community effort. Here you see some parts of the community. So the 4DN community, actually colleagues at UMass Medical School, the OMI community, the Bina and Quare Blini community, and developers of the tools and the funding. And I hope that this leaves us time to actually address some of the questions that might arise.